Hello there. Welcome to Quiet Science Channel. If you've found your way here tonight, perhaps you're settling in after a long day, looking for something calm and curious to accompany you as you drift towards sleep. Or maybe you're simply fascinated by the mysteries hidden within our own biology, the quiet processes that keep us alive and thinking and feeling. Either way, you're in the right place. Before we begin tonight's journey, if you find these gentle explorations helpful, if they give you a moment of peace or spark a question you hadn't considered, please consider liking this video and subscribing to the channel. It helps others discover these calm conversations about science, one quiet night at a time. Tonight, we're turning our attention to something you will do in just a few hours. Something you do every single day of your life, and yet something that remains, even now, one of biology's deepest mysteries: sleep. We spend roughly 1/3 of our lives asleep. That's about 25 years for someone who lives to 75. 25 years of lying still, eyes closed, vulnerable to the world, disconnected from our surroundings. It seems like an extraordinary waste of time. An organism that didn't need to sleep could spend those hours gathering food, building shelter, watching for danger, caring for offspring, exploring new territory. And yet every animal we know of sleeps, or does something very much like it. Mammals sleep, birds sleep, reptiles rest. Fish have periods of reduced activity. Even fruit flies and nematodes, creatures with nervous systems far simpler than ours, show something that looks unmistakably like sleep. They become still. They respond more slowly to their environment. And if you keep them awake, they later sleep longer and deeper to make up for it. This is the paradox at the heart of sleep. It seems dangerous, even foolish from an evolutionary standpoint. A sleeping animal cannot hunt, cannot mate, cannot defend its territory, cannot flee from predators. It is exposed, defenseless, oblivious. And yet sleep has persisted for hundreds of millions of years across countless species. Evolution does not preserve behaviors that harm survival. If sleep were merely a liability, it would have been eliminated long ago. So sleep must be doing something vital, something that cannot be accomplished while awake, no matter how still and quiet we remain. For most of human history, we had no idea what that something was. Sleep was simply rest, a time when the body and mind shut down to recover from the day's efforts. But over the past few decades, scientists have begun to uncover sleep's hidden purposes, and what they've found is far stranger and more essential than anyone imagined. Sleep is not a shutdown. It is not passive rest. During sleep, your brain is intensely active, replaying memories, solving problems, consolidating learning, processing emotions. Your brain is cleansing itself of toxic metabolic waste. Your neurons are resetting themselves to an optimal computational state. Your immune system is coordinating defenses. Your body is repairing tissue, balancing hormones, and preparing you for another day of wakefulness. Sleep is not one thing. It is many things happening at once. A symphony of biological processes that can only occur when consciousness releases its grip. Tonight, we'll explore what science has learned about why we sleep. We'll talk about the master clock in your brain that tells you when to feel drowsy and when to feel alert. We'll journey through the stages of sleep, from light drowsiness to the deepest slow wave sleep, and into the strange world of rapid eye movement sleep, where most dreaming occurs. We'll discover how sleep moves memories from temporary storage in the hippocampus to permanent archives in the cortex. We'll learn about the brain's waste clearance system, which becomes dramatically more active during sleep, flushing out proteins that would otherwise accumulate and cause dementia. We'll explore how REM sleep processes your emotions, separating the feeling from the memory, allowing you to remember experiences without being overwhelmed by them. We'll see how sleep restores your brain to an optimal state that physicists call criticality, a delicate balance that allows for flexible thinking and efficient information processing. And we'll confront the sobering reality of what happens when sleep is denied, how even a single night of lost sleep can impair your memory, judgment, and emotional control in ways that persist for days. This is a journey through one of neuroscience's greatest remaining mysteries. So let your breathing slow, let your body settle, and let's begin to understand why, after all these millions of years, we still need to close our eyes and drift into darkness each night. The universal mystery of sleep. If you look across the tree of life, sleep appears nearly everywhere. It is woven into the fabric of existence in ways that suggest it arose very early in evolutionary history and has been maintained ever since because the benefits outweigh the very real costs. Mammals, the group we belong to, show the most obvious forms of sleep. A dog curls up in its bed, muscles relaxed, breathing deep and rhythmic, eyes closed or half-lidded. The position itself signals vulnerability, a choice to trust the environment enough to let consciousness slip away. A lion sleeps for up to 20 hours each day, sprawled in the shade, conserving energy between hunts. This extended sleep makes sense for an apex predator, whose hunting strategy involves brief, intense bursts of activity, followed by long periods of rest and digestion. The lion's body cannot sustain constant activity, nor does it need to. Food, once caught, provides enough calories to last for days. An elephant sleeps standing up or lying down for just a few hours, always alert to threats, even in rest. Large herbivores face a different challenge. Their food, vegetation, is low in calories and requires nearly constant consumption. An elephant must eat for 16 to 18 hours each day to meet its metabolic needs. This leaves little time for sleep. Yet even elephants must sleep, suggesting that whatever sleep does, it cannot be skipped entirely, no matter how inconvenient. The most fascinating adaptations appear in marine mammals. Dolphins and whales face a unique challenge. If they fell completely unconscious underwater, they would drown. Unlike fish, they cannot extract oxygen from water. They must surface regularly to breathe, which requires some level of voluntary control even during rest. So their brains have evolved a remarkable solution. One hemisphere sleeps at a time while the other remains awake, allowing them to surface for air even during sleep. This is called unihemispheric sleep, and it reveals just how essential sleep is. Rather than eliminating it, evolution found a workaround. During unihemispheric sleep, one half of the dolphin's brain shows the slow waves characteristic of deep sleep, while the other half remains alert and active. The eye connected to the sleeping hemisphere closes, while the eye connected to the waking hemisphere stays open. After a period of time, the sides switch. The previously awake hemisphere falls asleep, and the previously sleeping hemisphere wakes up. This allows the dolphin to maintain awareness of its surroundings, navigate, surface for air, and even continue swimming in formation with its pod, all while obtaining the restorative benefits of sleep. Seals and sea lions also show unihemispheric sleep when in water, but when they haul out onto land, they switch to bilateral sleep, both hemispheres sleeping simultaneously, just like terrestrial mammals. This flexibility shows that unihemispheric sleep is not a fixed trait, but an adaptation deployed when circumstances demand it. The brain can choose its sleep mode based on environmental safety and physiological needs. Birds also sleep, and many show both NREM and REM sleep, just as mammals do. The similarity is remarkable given that birds and mammals evolved their large, complex brains independently after their evolutionary lineages split over 300 million years ago. This suggests that the selective pressures favoring complex sleep were strong enough to produce similar solutions in two very different groups of animals. Migratory birds face the same dilemma as marine mammals. During long flights that last days or even weeks, they cannot stop to rest. Some species have evolved the ability to engage in unihemispheric sleep while flying, keeping one eye open and one hemisphere awake to navigate and avoid collisions. Studies using miniature EEG devices attached to flying birds have confirmed this. The birds show slow wave activity in one hemisphere while the other remains active, and they switch sides periodically. Other migratory species may take brief micro-sleeps, moments of a few seconds where the brain dips into sleep-like states before snapping back to full alertness. These micro-sleeps are risky. A bird in micro-sleep is not fully aware of its surroundings and could collide with obstacles or lose altitude. Yet the pressure to sleep is so great that birds accept this risk rather than staying fully awake for the entire journey. Interestingly, some studies have found that migratory birds sleep less during migration than during other times of the year, but they compensate by sleeping more intensely before and after the journey. They appear to build up a reserve of rest and then pay back the sleep debt once they reach their destination. This suggests that while sleep can be reduced temporarily, it cannot be eliminated. The need accumulates and must eventually be satisfied. Non-migratory birds show sleep patterns more similar to mammals, with clear periods of NREM and REM sleep, though REM episodes in birds tend to be much shorter, often lasting only a few seconds compared to the minutes-long REM periods in mammals. Songbirds have proven particularly interesting for sleep research because they learn their songs through a process analogous to human language learning, and this learning is enhanced by sleep. Young birds that practice their songs during the day show neural replay of the song patterns during sleep, much like the memory replay seen in mammalian brains. Reptiles rest, though whether they experience sleep stages comparable to mammals remains debated and fascinatingly complex. Lizards and turtles show periods of behavioral quiescence with reduced responsiveness to stimuli. Their eyes close, their bodies become still, and they withdraw to safe locations. This looks like sleep, but the brain activity is different from what we see in sleeping mammals. Some studies have found EEG signatures in reptiles that resemble mammalian sleep, including something that might be precursors to REM sleep, but the evidence remains incomplete and contentious. A study of bearded dragons, a species of Australian lizard, found patterns of brain activity during rest that bore striking similarities to mammalian REM and slow-wave sleep, including rapid eye movements and changes in EEG frequencies. This was surprising because reptiles and mammals diverged evolutionarily before these sleep states were thought to have emerged. If reptiles truly have REM-like states, it suggests that either REM sleep evolved much earlier than previously believed, perhaps in the common ancestor of all amniotes, or that similar sleep states evolved independently in different lineages through convergent evolution. Both possibilities are fascinating and tell us something important about the fundamental nature of sleep. The debate continues because not all reptile species show these patterns, and the sleep of reptiles is highly temperature dependent. Reptiles are ectothermic, meaning they rely on external sources for body heat. When temperatures drop, their metabolic rate slows, and they enter states of torpor that look like sleep, but may be fundamentally different. The question becomes, is a cold, torpid reptile truly sleeping, or is it simply inactive due to insufficient energy to move? The distinction matters for understanding what sleep is at its core. Fish exhibit clear rest periods that meet the behavioral criteria for sleep. Zebrafish, a common laboratory model, become still at night, settling to the bottom of the tank with reduced muscle tone and slower gill movements. Their responsiveness to external stimuli drops significantly. If you flash a light or create a vibration, a resting zebrafish takes longer to respond than an active one. If you prevent them from resting by gently disturbing them throughout the night, they show rebound sleep the next night, sleeping longer and deeper to compensate. This suggests genuine sleep, not merely inactivity. Different fish species have evolved diverse sleep strategies adapted to their ecological niches. Some species of sharks must keep swimming to force water over their gills for respiration, so they engage in periods of reduced activity while continuing to move slowly forward. Other fish, like parrotfish, secrete a mucus cocoon around themselves at night, which may serve as protection from predators while they sleep. The cocoon takes time to produce and dissolve, suggesting that the fish views sleep as valuable enough to invest this effort. Insects sleep. Fruit flies have a sleep-like state where they become immobile. Their responsiveness drops, and they require more intense stimuli to wake. This was discovered through careful behavioral observations. Researchers noticed that fruit flies have periods of prolonged inactivity, usually at night in a dark environment, where they remain completely still for minutes or hours at a time. If you gently tap the container, a sleeping fly does not react. A stronger tap might cause it to stir briefly before settling back into stillness. Only a vigorous disturbance will fully wake it. If you keep fruit flies awake by gently shaking them or using other disturbances throughout the night, they accumulate sleep debt and later sleep more to recover. They sleep longer and more deeply the following day, showing the hallmark rebound effect of true sleep. Genetic studies have identified numerous genes in fruit flies that regulate sleep, and many of these genes have equivalents in mammals that play similar roles. This suggests deep evolutionary conservation of sleep mechanisms. The same appears true for bees, ants, and other insects. Honeybees show clear rest periods where they become immobile in the hive, often with drooping antennae, a reliable sign that they are not merely resting, but sleeping. Younger bees sleep less than older bees, and forager bees that have spent the day flying long distances to collect pollen and nectar sleep more intensely than bees that remained in the hive. This suggests that sleep in bees, as in mammals, is linked to the metabolic demands of activity. Even creatures with brains containing just a few 100,000 neurons, rather than the 86 billion in a human brain, still need to sleep. This tells us something profound. Sleep is not a luxury afforded only to complex nervous systems. It is a fundamental requirement for neural tissue, period. Perhaps most remarkably, sleep has been observed in cyanidarians, a group that includes jellyfish, corals, and sea anemones. These animals diverged from other animals over 600 million years ago, long before brains as we know them evolved. They have diffuse nerve nets rather than centralized brains, yet they still sleep. A species of upside-down jellyfish, Cassiopeia, shows a clear behavioral state that meets the criteria for sleep. These jellyfish spend most of their time resting upside down on the seafloor, pulsing their bells rhythmically to draw water and food particles over their tentacles. At night, the rate of pulsing slows significantly. The jellyfish become less responsive to stimuli. A gentle water current that would normally provoke a change in pulsing rate has little effect during this rest state. And crucially, if you deprive them of this rest by disturbing them throughout the night, they show a compensatory increase in the resting behavior the next day, pulsing even more slowly and for longer periods. This discovery was astonishing. It suggests that sleep, or something very much like it, may have originated in the common ancestor of all animals with nervous systems, perhaps as a way to manage the metabolic demands of neural tissue. Neurons are expensive. They consume enormous amounts of energy, and the waste products of their activity must be managed. Perhaps even the simplest nervous systems, nerve nets with no centralized brain, require periods of reduced activity to maintain function. If so, sleep may be as old as neurons themselves, a constraint built into the very architecture of neural computation. Why has sleep persisted so universally? One major theory is energy conservation. Staying alert and responsive requires enormous metabolic resources, especially for the brain. The human brain, while representing only 2% of body weight, consumes 20% of the body's total energy at rest. During intense cognitive activity, this demand increases further. If an animal's environment becomes less favorable for activity, either because of darkness, temperature, or the absence of food, it makes sense to enter a low-energy state. Sleep achieves this. During sleep, metabolic rate drops by up to 15%, body temperature decreases slightly, and energy is conserved. For small animals with high metabolic rates, like hummingbirds and bats, this conservation can be critical to survival. Some hummingbirds enter a torpor-like state during sleep, where their body temperature plummets from around 41 degrees Celsius during the day to as low as 18 degrees Celsius at night, saving precious calories that would otherwise be burned, maintaining their tiny, hyperactive bodies. Without this energy-saving adaptation, a hummingbird might starve overnight, unable to maintain its metabolic furnace until the next opportunity to feed. Torpor during sleep allows it to stretch its energy reserves through the long hours of darkness. Bats, similarly, can enter torpor during their daytime sleep, reducing their body temperature and metabolic rate dramatically. This is especially important for insectivorous bats in temperate regions, where prey availability fluctuates seasonally. By entering deep torpor during sleep, these bats can survive periods when food is scarce, emerging only when conditions improve. But energy conservation alone does not fully explain sleep. If conservation were the only goal, animals could simply rest quietly while remaining awake and vigilant. A rabbit sitting motionless in its burrow, ears swiveling to catch sounds of predators, conserves energy almost as effectively as a sleeping rabbit, yet sleep still happens. The rabbit could, in principle, remain awake but inactive, conserving energy while maintaining awareness. But it does not. Instead, it falls asleep, entering a state of reduced consciousness where it is vulnerable. This tells us sleep serves purposes beyond mere inactivity. Another evolutionary perspective focuses on the niche separation theory, sometimes called the adaptive inactivity theory. During a 24-hour cycle, the environment changes dramatically. Day brings light, warmth, and the activity of diurnal predators and prey. Night brings darkness, cold, and the stirring of nocturnal hunters. An animal cannot be perfectly adapted to both environments. A visual system optimized for daylight vision performs poorly in darkness. A metabolism tuned for warm temperatures struggles in cold. Trying to be active all the time would mean functioning poorly in at least one of these environments, giving an advantage to competitors more specialized for that niche. Sleep, according to this view, is evolution's way of forcing an animal to pick one niche and commit to it. Diurnal animals sleep at night, removing themselves from a dangerous environment they are poorly equipped to navigate. Their eyes cannot see well in darkness. Their predators are active. Their prey are hidden, and the cold saps their energy. Better to sleep through the night, conserving energy and avoiding danger, than to blunder about ineffectively. Nocturnal animals sleep during the day for similar reasons, avoiding the heat and the predators they cannot see as well in sunlight. Sleep ensures that animals remain in their optimal niche, even if they feel restless or curious about the other half of the day. This theory gains support from observations of sleep in dangerous environments. Animals that are highly vulnerable to predation tend to sleep in short bouts, waking frequently to check for danger. Prey animals like rabbits and deer sleep far less than predators like lions and wolves, and their sleep is more fragmented. They cannot afford deep, uninterrupted sleep when threats surround them. Predators, by contrast, face fewer threats and can sleep more deeply and for longer periods. Yet even this explanation feels incomplete when examined closely. Sleep is not simply hiding away in a safe place. It involves specific brain states, precise patterns of neural activity, and biochemical changes that appear to serve particular functions. If sleep were only about niche separation and avoiding danger, why would the brain be so busy during it? Why would memories be replayed, emotions processed, and waste cleared specifically during sleep, rather than during quiet waking rest in a safe burrow? A rabbit hiding quietly in its burrow while awake could theoretically accomplish the goals of energy conservation and niche separation. But the rabbit sleeps instead, entering a state where its brain engages in activities that appear impossible during wakefulness. This suggests that the real answer lies not in what sleep allows animals to avoid, but in what sleep allows the brain to accomplish. The answer, we now understand, is that sleep is polyfunctional. It does not have a single purpose, but many purposes, each critical in its own way. Sleep conserves energy, yes. Sleep removes us from dangerous times and places, yes. But sleep also allows the brain to do things it cannot do while awake. Things that require a fundamentally different mode of operation. And that is where the story becomes truly fascinating, because to understand those things, we need to understand how sleep is regulated. And that begins with a tiny cluster of neurons deep in your brain. The master clock within your body runs on rhythms, not just the beating of your heart or the rise and fall of your breath, but slower rhythms that repeat over the course of about 24 hours. These are called circadian rhythms, from the Latin circadium, meaning approximately a day. Nearly every cell in your body contains a biological clock, a set of genes that turn each other on and off in a self-sustaining cycle that takes roughly a day to complete. But these cellular clocks do not run independently. They are synchronized by a master clock, a conductor that ensures all the body's rhythms play in harmony. That master clock resides in a structure called the suprachiasmatic nucleus, or SCN. The SCN is tiny, a pair of clusters containing about 20,000 neurons in total, nestled just above the optic chiasm, the point where the optic nerves from each eye cross over in the brain. This location is not accidental. The SCN receives direct input from specialized photoreceptive cells in the retina, cells that are not the rods and cones responsible for vision, but rather ganglion cells containing a photopigment called melanopsin. These cells detect the overall light levels in the environment and send that information directly to the SCN. When light enters your eyes in the morning, these cells tell the SCN it is day. When darkness falls, they signal night. The SCN uses this information to synchronize the body's internal rhythms with the external world. Without this entrainment, your circadian clock would drift. In experiments where people are placed in environments with no time cues, no windows, no clocks, no sunrise or sunset, their sleep-wake cycles begin to free run. Instead of following a 24-hour schedule, most people settle into a rhythm closer to 24.25 hours, sometimes even longer. Each day, they go to bed a little later and wake a little later than the day before. Over time, their internal day drifts out of sync with the actual day outside. This demonstrates that the circadian clock is not precisely 24 hours. It is approximately 24 hours, and it needs daily resetting by light to stay aligned with the rotation of the Earth. The SCN accomplishes this alignment through a network of neural and hormonal signals. During the day, the SCN suppresses the production of melatonin, a hormone synthesized by the pineal gland. Melatonin is sometimes called the hormone of darkness. Its levels are very low during daylight hours. As evening approaches and light levels drop, the inhibitory signals from the SCN diminish, and melatonin production ramps up. Melatonin levels rise through the night, peaking in the early morning hours before dawn, then falling again as sunrise approaches. This nightly surge of melatonin helps promote sleepiness and signals to the body that it is time to rest. But melatonin is not the only player. The SCN also influences body temperature, which follows a circadian rhythm of its own. Core body temperature is lowest in the early morning, around 4 or 5 a.m., and highest in the late afternoon or early evening. This rhythm is synchronized with sleep. The drop in body temperature in the evening is part of what makes you feel drowsy. Conversely, the rise in temperature in the morning helps promote wakefulness. Disrupting this rhythm, for instance, by taking a very hot bath right before bed, can initially make it harder to fall asleep, though the subsequent cooling may help. The SCN coordinates dozens of other rhythms. Cortisol, the stress hormone, follows a daily cycle, surging in the early morning to help wake you up and prepare you for the day. Growth hormone is released predominantly during deep sleep. Appetite-regulating hormones like ghrelin and leptin follow circadian patterns, which is one reason why disrupted sleep often leads to changes in hunger and weight. Blood pressure, immune function, cell division, even the sensitivity of your sense of smell, all vary across the day and night, coordinated by the SCN's pervasive influence. This coordination is essential for health. When the SCN is damaged, animals lose their clear sleep-wake rhythms. They still sleep, but the sleep becomes fragmented, scattered across the day and night in short bursts rather than consolidated into a single long period. In humans, similar disruptions occur with disorders like irregular sleep-wake rhythm disorder, often seen in people with advanced dementia. The SCN degenerates, and the rhythms fall apart. Circadian rhythm disruption is also a major problem for shift workers, people who work at night and try to sleep during the day. The SCN is designed to promote wakefulness during daylight and sleep during darkness. Fighting against this system leads to chronic misalignment. Shift workers often report poor sleep quality even when they have adequate time to sleep, because their circadian clock is telling them to be awake. They also face higher risks of cardiovascular disease, metabolic disorders, mood disturbances, and certain cancers, likely due to the chronic stress of circadian disruption. Jet lag is a temporary version of the same problem. When you fly across multiple time zones, your internal clock does not instantly adjust. For several days, your SCN is still following the schedule of your departure city, while your body is in a new time zone. You feel drowsy when you should be alert and wide awake in the middle of the night. The SCN gradually adjusts, usually at a rate of about 1 time zone per day. But until it does, the mismatch between internal and external time is uncomfortable and impairing. Yet the circadian clock alone does not fully determine when you sleep. There is a second process at work, a process called sleep homeostasis. If the circadian clock is like a timer, telling you when it is an appropriate time to sleep, sleep homeostasis is like a pressure gauge, telling you how much you need to sleep. The longer you stay awake, the more sleep pressure builds. This pressure appears to be driven by the accumulation of adenosine, a molecule that is a byproduct of cellular energy use. Neurons are constantly burning ATP, the cell's energy currency, and when ATP breaks down, it releases adenosine. Adenosine accumulates in the brain throughout the day, binding to receptors that slow neural activity and promote feelings of tiredness. Caffeine works by blocking adenosine receptors, temporarily preventing this sleepiness signal. During sleep, adenosine levels drop. The brain clears the accumulated adenosine, and you wake feeling refreshed with the sleep pressure reset to 0. If you do not sleep, adenosine continues to build, and the pressure to sleep becomes overwhelming. People who are severely sleep-deprived often experience microsleeps, brief moments where the brain dips into sleep for a few seconds, even while the person is ostensibly awake. These microsleeps are not voluntary. They are the brain forcing itself to rest when sleep pressure becomes unbearable. The interplay between the circadian clock and sleep homeostasis creates your daily pattern of alertness and drowsiness. In the late afternoon, your circadian clock promotes wakefulness, even though some sleep pressure has built, which is why most people feel a second wind in the evening. But as night falls and melatonin rises, and as sleep pressure continues to mount, both systems align to promote sleep. You drift off, and the night's work begins. The architecture of sleep is not a single uniform state. It is a progression through distinct stages, each with its own brain activity patterns, physiological characteristics, and functions. When you fall asleep, your brain does not simply shut off. It transitions through a carefully choreographed sequence of states that repeat in cycles throughout the night. Scientists measure sleep using polysomnography, a technique that simultaneously records electrical activity in the brain using electroencephalography, or EEG, eye movements using electrooculography, or EOG, and muscle tone using electromyography, or EMG. Together, these measurements allow sleep to be divided into stages. There are 2 broad categories of sleep: non-rapid eye movement sleep, or NREM, and rapid eye movement sleep, or REM. NREM sleep is further divided into 3 stages: N1, N2, and N3. N3 is often called slow wave sleep or deep sleep, and it is the stage from which it is hardest to be woken. When you first lie down and close your eyes, you enter stage N1. This is the lightest stage of sleep, the transition between wakefulness and sleep. EEG shows a slowing of brain waves from the rapid beta waves of alert wakefulness to slower alpha and theta waves. Your muscles relax, your breathing slows, and your awareness of the outside world dims. If someone were to wake you from N1, you might not even realize you had been asleep. You might say you were just resting. N1 typically lasts just a few minutes. Stage N2 follows. EEG now shows two characteristic features: sleep spindles and K-complexes. Sleep spindles are brief bursts of fast oscillatory activity, sharp peaks of brain waves that last a second or so. K-complexes are sudden, large spikes in the EEG, brief deflections that stand out against the background rhythm. The exact function of spindles and K-complexes remains debated, but they appear to play roles in protecting sleep from disruption by external stimuli and in memory consolidation. Stage N2 is still relatively light sleep, and it makes up a large proportion of the night, perhaps 40 to 50% of total sleep time. After some time in N2, the brain descends into stage N3, slow wave sleep. This is deep sleep, the restorative sleep that leaves you feeling refreshed. EEG shows large, slow delta waves, oscillations at less than 4 cycles per second. These waves are much larger and slower than anything seen during wakefulness or lighter sleep. Brain activity becomes highly synchronized, with vast numbers of neurons firing in rhythmic unison. This synchrony is thought to be crucial for the consolidation of certain types of memory, particularly declarative memories, facts and events that can be consciously recalled. During slow wave sleep, your heart rate and breathing slow to their lowest levels. Blood pressure drops. Growth hormone is released in pulses, particularly in children and adolescents, aiding tissue repair and growth. It is difficult to wake someone from slow wave sleep. If you are woken from it, you feel groggy and disoriented, a state called sleep inertia that can last for many minutes. After a period of slow wave sleep, the brain does something unexpected. Instead of deepening further into unconsciousness, it becomes active again, but in a different way. You enter REM sleep. REM sleep is unlike any other state of consciousness. EEG during REM resembles wakefulness, with fast, desynchronized activity. The brain is busy, almost as active as when you are awake. Yet, you are deeply asleep, disconnected from your environment, and your body is paralyzed. This paralysis is called atonia, and it is mediated by neurons in the brainstem that actively inhibit motor neurons, preventing your muscles from moving. Only the muscles controlling eye movement and breathing are spared. Your eyes dart rapidly back and forth beneath your closed eyelids, giving REM sleep its name. Your breathing becomes irregular, sometimes rapid, sometimes slow, and your heart rate and blood pressure fluctuate. REM sleep is the stage where most vivid dreaming occurs. If someone is woken from REM, they are much more likely to report detailed, bizarre, emotionally intense dreams than if woken from NREM sleep. Dreams from NREM sleep tend to be more thought-like, less visual, less narrative. But REM dreams are often cinematic, filled with vivid imagery, strange juxtapositions, impossible events, and strong emotions. The brain's neurochemistry during REM is unique. Activity in the brainstem structures that release norepinephrine and serotonin, two major neurotransmitters, drops to near zero. This creates a brain environment that is rich in acetylcholine but poor in monoamines. This neurochemical signature appears to be important for the functions of REM sleep, allowing certain types of neural plasticity that cannot occur during waking or NREM sleep. The first REM period of the night is short, usually lasting just 5 to 10 minutes. As the night progresses, REM periods grow longer, and the final REM period before waking may last half an hour or more. Meanwhile, slow-wave sleep dominates the first half of the night and diminishes in the second half. A full sleep cycle, from the start of N1 through slow-wave sleep and into REM, lasts about 90 to 110 minutes. Most people go through 4 to 6 cycles per night. This cycling is not arbitrary. Each stage appears to serve different functions, and the pattern of cycling ensures that the brain gets adequate time in each state. As we age, sleep architecture changes. Newborns spend about half their sleep time in REM, possibly because REM sleep plays a role in brain development and synapse formation. By adulthood, REM makes up about 20 to 25% of sleep. Older adults often experience less deep sleep and more frequent awakenings. The amount of slow-wave sleep declines with age, which may contribute to memory difficulties and reduced physical restoration in elderly people. The architecture of sleep reveals that rest is not passive. It is an organized, dynamic process, a nightly journey through states that serve different purposes. And as we will see, each stage contributes in its own way to the brain's health and function. Memory's nighttime theater. One of the most well-established functions of sleep is memory consolidation, the process by which newly learned information is stabilized and integrated into long-term storage. Without sleep, memories remain fragile and easily disrupted. With sleep, they become robust and enduring. The story begins in the hippocampus, a seahorse-shaped structure deep in the temporal lobe of the brain. The hippocampus is essential for forming new episodic memories, memories of events and experiences. When you learn something new, a new face, a new route through the city, a new piece of information, the hippocampus encodes that experience by creating patterns of neural activity. These patterns are specific to that experience. A particular set of neurons fires in a particular sequence, representing what happened. But the hippocampus has limited capacity. It cannot store everything forever. It is more like a temporary buffer, a sketch pad where new information is held until it can be transferred to long-term storage in the cortex, the vast outer layers of the brain. This transfer happens during sleep, in a process that is both elegant and essential. During slow-wave sleep, the hippocampus becomes highly active in specific patterned ways. Neurons that fired during a learning experience reactivate in the same sequence, replaying the experience at high speed. These replay events, often called sharp wave ripples because of their distinctive appearance on EEG recordings, occur hundreds or thousands of times throughout the night. Each replay is a rapid recapitulation of the day's experiences, compressed into a fraction of a second, sometimes playing forward, sometimes backward, sometimes in fragments. Imagine you spent the day learning a new route through your city. As you navigated, specific neurons in your hippocampus fired in a particular sequence, each representing a landmark or turn. That night, during slow-wave sleep, those same neurons fire again in the same order, replaying your journey. The replay is much faster than the original experience, compressed from minutes into milliseconds. The brain is essentially practicing the route over and over, strengthening the neural connections that represent it. Why does the brain do this? The leading theory is that replay allows the hippocampus to teach the cortex. Each time the hippocampus replays a memory, it sends signals to the cortex, gradually strengthening the connections there that represent the same information. Over many replays, over many nights, the cortical representation becomes strong enough to stand on its own. the memory no longer depends on the hippocampus. It has been consolidated into long-term storage, becoming part of the permanent record of your life. This process is called systems consolidation, and it appears to be one of the primary functions of slow-wave sleep. The slow oscillations that characterize this stage, those large rhythmic waves visible on EEG, create windows of opportunity for communication between the hippocampus and cortex. The oscillations have two phases: up states, when neurons are active and firing, and down states, when neurons are quiet and hyperpolarized. During the up states of the oscillation, when neurons are firing, replay events occur. The hippocampus broadcasts its memories to the cortex. During the down states, when neurons are quiet, the cortex can integrate the information it just received, adjusting synaptic weights and forming new connections without interference. This alternation between information transmission and integration appears to be crucial for effective learning. The coordination is remarkable. Sleep spindles, those brief bursts of fast oscillatory activity seen during stage N2, are often synchronized with sharp wave ripples and slow oscillations. The three rhythms nest together, creating a precisely timed sequence that facilitates memory transfer. Disrupting any one of these rhythms disrupts consolidation. Evidence for sleep's role in memory is overwhelming and comes from multiple lines of research. Studies have shown that people who sleep after learning perform better on memory tests than people who stay awake, even when the total time between learning and testing is the same. This demonstrates that sleep is not simply protecting memories from interference, though it does that too. Sleep actively enhances them through consolidation processes that only occur during sleep. One classic experiment had participants learn pairs of words in the morning. Half the group then stayed awake all day, while the other half took a nap. When both groups were tested in the evening, the nappers recalled significantly more word pairs than the non-nappers, despite both groups having the same amount of time pass between learning and testing. The only difference was sleep. Even more remarkably, people sometimes recall more information after sleep than they did immediately after learning, a phenomenon called sleep-dependent memory enhancement. In studies where people are tested immediately after learning, then again after sleeping, their scores often improve. This is counterintuitive. Normally, memory degrades with time, but sleep reverses this trajectory, actually strengthening memories rather than allowing them to fade. Brain imaging studies have revealed the neural basis of this enhancement. When people learn new information and then sleep, functional MRI shows increased connectivity between the hippocampus and various cortical regions during slow-wave sleep. The more connectivity increases, the better the memory performance the next day. This suggests that the physical pathways linking the hippocampus and cortex are being strengthened during sleep through repeated reactivation. Different types of memories benefit from different stages of sleep, revealing that sleep is not a one-size-fits-all process. Declarative memories, the facts and events you can consciously recall, things like what you ate for breakfast yesterday or the capital of France, benefit most from slow-wave sleep. Studies that selectively enhance slow-wave sleep, using techniques like acoustic stimulation that boosts the amplitude of slow oscillations, find improved declarative memory consolidation. Procedural memories, the skills and habits that are performed without conscious thought, like riding a bicycle, typing on a keyboard, or playing a musical instrument, benefit more from REM sleep and stage 2 NREM sleep, particularly the periods rich in sleep spindles. Musicians who practice a new piece before sleep show improved performance after a night's rest, and the degree of improvement correlates with the amount of REM sleep and spindle density they obtained. Their fingers, it seems, practice in their sleep, refining the motor sequences without conscious effort. But memory consolidation is not just about strengthening memories. It is also about selecting which memories to keep and which to let fade. The brain cannot store everything. You do not need to remember every mundane detail of every moment. Sleep appears to help with this triage, strengthening memories that are emotionally salient or useful, and allowing irrelevant memories to weaken. Recent research has also uncovered a fascinating resetting process that happens during sleep. A study published in 2024 found that during deep sleep, certain areas of the hippocampus go completely silent for brief periods. This silencing appears to reset the neurons, clearing them of the day's activity and preparing them to encode new memories the next day. Without this reset, the hippocampus would become saturated, unable to take in new information. Sleep, in this view, is not just about consolidating old memories, but also about making room for new ones. The dialogue between hippocampus and cortex during sleep is not one way. The cortex also sends signals back to the hippocampus, providing context and helping to integrate new memories with existing knowledge. This bidirectional communication allows the brain to build abstract representations, to extract the gist of experiences rather than storing them as isolated episodes. It is how you learn patterns, rules, and concepts rather than just remembering individual events. This process is particularly important for creativity and problem solving. There are countless anecdotes of people waking with solutions to problems that baffled them the day before. The chemist Auguste Kekule claimed he discovered the ring structure of benzene after dreaming of a snake biting its own tail. The artist Salvador Dalí used to hold a key above a metal plate as he drifted to sleep, allowing the key to drop and wake him at the moment of transition, capturing hypnagogic imagery for his paintings. While these stories are colorful, modern science has shown that sleep genuinely aids insight. Studies where people are given puzzles before sleep often find that they solve the puzzles more readily after waking, suggesting that sleep allows the brain to explore different combinations of ideas, to form new connections between seemingly unrelated concepts. This creative enhancement appears to happen primarily during REM sleep, where the brain's activity is more chaotic and less constrained. The lack of norepinephrine, the reduction in logical control from the prefrontal cortex, and the heightened activity in associative brain regions all combine to allow freer exploration of mental space. Dreams may be the conscious reflection of this process, the mind wandering through novel configurations of thought. Yet sleep's role in memory extends beyond consolidation and creativity. Sleep also appears to help with forgetting, specifically with forgetting the emotional intensity of memories while retaining their factual content. This is a subtle but profoundly important function, and it happens primarily during REM sleep, a topic we will return to shortly. For now, it is enough to understand that every night your brain replays the day. It sifts through experiences, selects what matters, strengthens connections, integrates knowledge, resets for the next day, and in doing so, constructs the continuity of your life, the sense that you are a person with a past, a present, and a future. Without sleep, this continuity fragments, memories become hazy, learning becomes difficult, the past does not settle into place, and the mind struggles to move forward. The brain's nightly wash cycle. The brain is a remarkably active organ. Despite accounting for only about 2% of the body's weight, it consumes roughly 20% of the body's energy. This intense metabolism produces waste. Cells break down nutrients, synthesize proteins, and generate byproducts that must be cleared away. In the body, the lymphatic system performs this waste removal, carrying away cellular debris and toxins through a network of vessels. But the brain does not have a traditional lymphatic system. For years, scientists wondered how the brain stayed clean. The answer came in 2012 with the discovery of the glymphatic system, a term coined by neuroscientist Maiken Nedergaard and her colleagues. The name combines glial, referring to the brain's supportive cells, with lymphatic, acknowledging its functional similarity to the body's waste clearance network. The glymphatic system works like this. Cerebrospinal fluid, or CSF, the clear liquid that surrounds the brain and spinal cord, flows into the brain along pathways surrounding blood vessels, spaces called perivascular spaces. This influx of CSF is driven by the pulsation of arteries with each heartbeat. The CSF enters the brain tissue, where it mixes with interstitial fluid, the fluid between brain cells. As it mixes, the CSF picks up metabolic waste products, including proteins that have accumulated during waking activity. One of the most important waste products cleared this way is beta amyloid, a protein fragment that forms sticky plaques in the brains of people with Alzheimer's disease. Beta amyloid is produced constantly by neurons during normal activity, and in healthy brains, it is cleared before it can accumulate. But if clearance is impaired, beta amyloid builds up, forming plaques that disrupt neural function and contribute to cognitive decline. The glymphatic system relies on specialized channels called aquaporin 4, or AQP4 water channels. These channels are embedded in the membranes of astrocytes, star-shaped glial cells, whose foot processes wrap around blood vessels. AQP4 channels facilitate the rapid movement of fluid between the perivascular space and the brain tissue. Animals lacking AQP4 show dramatically reduced glymphatic clearance and increased accumulation of beta amyloid. What does all this have to do with sleep? It turns out that glymphatic clearance is dramatically enhanced during sleep compared to wakefulness. Studies in mice have shown that the volume of the extracellular space, the gaps between brain cells, increases by up to 60% during sleep. This expansion reduces resistance to fluid flow, allowing CSF to move more freely and flush out waste more efficiently. During wakefulness, the extracellular space is more constricted, and clearance drops to a fraction of its nighttime rate. The mechanism behind this expansion involves the neurotransmitter norepinephrine, also known as noradrenaline. During wakefulness, neurons in a brainstem structure called the locus coeruleus release norepinephrine throughout the brain. Norepinephrine keeps you alert, focused, and responsive. But it also causes the extracellular space to contract, likely by triggering changes in the shape and volume of glial cells. During non-REM sleep, norepinephrine levels plummet. The locus coeruleus falls silent. Without norepinephrine, the extracellular space expands and glymphatic clearance increases. A study published in January 2025 used a technique called fiber photometry to measure norepinephrine levels in real time in sleeping mice. The researchers found that norepinephrine oscillates rhythmically during non-REM sleep, and these oscillations drive corresponding pulses of blood volume and CSF flow. The alternating constriction and dilation of cerebral arteries acts like a pump, propelling CSF through the brain. Importantly, not all sleep is equal for glymphatic function. The deepest stages of non-REM sleep, when slow waves dominate, appear to be the most effective for waste clearance. REM sleep, with its elevated norepinephrine levels and brain activity resembling wakefulness, shows less glymphatic activity. This suggests that one function of slow-wave sleep is specifically to allow the brain to cleanse itself. The implications for health are profound. People who chronically sleep poorly, who do not get enough slow-wave sleep, may accumulate more beta amyloid and other waste products over time. Studies have found that even a single night of sleep deprivation leads to measurable increases in beta amyloid in the brain. Chronic sleep deprivation is associated with higher rates of Alzheimer's disease and other forms of dementia. The relationship appears to be bidirectional. Poor sleep leads to amyloid accumulation, and amyloid accumulation disrupts sleep, creating a vicious cycle that accelerates cognitive decline. Sleep position may even matter. One study found that glymphatic clearance is most efficient when animals sleep on their side, less efficient when sleeping on the back, and least efficient when sleeping face down. The reasons are not entirely clear, but it may relate to how gravity affects CSF flow and venous drainage. In 2024, scientists provided the first direct confirmation of the glymphatic system in humans. Researchers at Oregon Health and Science University used imaging techniques during neurosurgery to track the movement of a contrast agent injected into the cerebrospinal fluid of 5 patients. The images revealed the contrast flowing along perivascular spaces, exactly as predicted by the glymphatic model. This was the first time the waste clearance pathways in the human brain were visualized directly. More recently, researchers have shown that neuronal activity itself influences glymphatic flow. Synchronized neural oscillations, the rhythmic firing patterns that occur during sleep, appear to stimulate the rhythmic constriction and dilation of blood vessels, enhancing the pumping action that drives CSF through the brain. This adds another layer to the relationship between sleep and waste clearance. It is not just that sleep allows clearance, but that the specific patterns of brain activity during sleep actively promote it. This discovery helps explain why sleep is so restorative. You are not simply resting when you sleep. Your brain is actively engaged in maintenance, washing away the metabolic debris that accumulated during waking hours. It is a process that cannot be fully replicated during quiet waking rest, because the necessary changes in brain state, in norepinephrine levels, in extracellular space volume, in synchronized oscillations, only happen during true sleep. The glymphatic system is still being studied, and many questions remain. Do all brain regions clear waste at the same rate? Are there other waste products besides beta amyloid that are critical? Can glymphatic function be enhanced to treat or prevent neurodegenerative diseases? But the fundamental insight is clear. Sleep is the brain's dishwasher, and without it, the accumulated grime of waking life begins to interfere with the machinery of thought. Emotional alchemy in REM sleep. Emotions shape our lives. They guide our decisions, color our perceptions, and bind us to other people. But emotions can also overwhelm us, particularly when tied to traumatic or intensely negative experiences. A single frightening event can leave an emotional imprint that persists for years, triggering anxiety, avoidance, and distress whenever the memory is recalled. This is the essence of post-traumatic stress disorder, a condition where the emotional charge of a memory remains unbearably high. For most of us, though, the emotional intensity of experiences fades with time. You can remember a painful breakup or a terrifying accident without reliving the full force of the original emotion. The memory remains, but the feeling softens, This is not merely the passage of time. It is an active process, and it happens during sleep, specifically during REM sleep. REM sleep has a unique neurochemistry. As mentioned earlier, the brainstem nuclei that release norepinephrine and serotonin fall silent during REM. This is unlike any other state of consciousness, waking or sleeping. The brain is active, processing information, experiencing vivid dreams, yet it operates in an environment almost devoid of monoamines. Norepinephrine, also called adrenaline in the body, is the brain's stress chemical. It sharpens focus, heightens arousal, and amplifies emotional responses. When you experience something frightening or exciting, norepinephrine surges. Its presence during memory encoding tags that memory as emotionally significant. Later, when the memory is recalled, norepinephrine can resurge, bringing back the emotional intensity. But during REM sleep, norepinephrine is absent. This creates an opportunity. Emotional memories can be reactivated and processed in the hippocampus and cortex without the accompanying surge of stress chemicals. The memory is replayed, but the emotion is muted. The brain, in effect, learns to separate the factual content of the memory from its emotional tone. You remember what happened, but the sting is reduced. This process is sometimes called emotional depotentiation, and it may be one of the most important functions of REM sleep. Neuroscientist Matthew Walker and his colleagues have proposed that REM sleep serves as a form of overnight therapy, a time when the brain reprocesses emotional experiences in a safe neurochemical environment. The vivid, often bizarre nature of dreams may reflect this reprocessing, the brain weaving emotional memories into strange narratives as it works to integrate and diffuse them. Studies support this idea. When people are shown emotionally upsetting images and then tested later, those who sleep, particularly those who get adequate REM sleep, show reduced emotional reactivity to the images compared to those who stayed awake. The images are recognized, the memory is intact, but the emotional response is dampened. Brain imaging studies show that the amygdala, the brain's emotional alarm center, becomes less reactive to previously seen emotional stimuli after a night of sleep with REM. Conversely, sleep deprivation, particularly REM deprivation, leaves people emotionally raw. Even minor stressors provoke outsized reactions. The prefrontal cortex, which normally regulates the amygdala and provides context and control to emotional responses, loses its grip. The amygdala becomes hyperactive, responding excessively to negative stimuli. People become irritable, anxious, and prone to emotional outbursts. Dreams, the conscious experience of REM sleep, often have strong emotional content. People report fear, joy, sadness, anger, confusion, all experienced vividly, even though the events in the dream are impossible or nonsensical. About 75 to 95% of dream reports contain emotional themes. The content of dreams is not usually a direct replay of the day's events. Instead, dreams seem to pick up on the emotional themes of recent experiences and weave them into new scenarios. You might not dream about the argument you had, but you might dream about being chased or lost or humiliated, capturing the emotional essence without the literal details. This thematic representation suggests that REM sleep is not just replaying memories, but actively processing them, extracting their emotional significance, and integrating it with other memories and concerns. In this way, REM sleep contributes to emotional regulation and mental health. It is no coincidence that nearly every psychiatric disorder involving emotional dysregulation, depression, anxiety disorders, PTSD, bipolar disorder, is also associated with disrupted REM sleep. In PTSD, the normal function of REM sleep appears to break down. Instead of depotentiating the traumatic memory, REM sleep reactivates it with full emotional force, producing nightmares that feel like reliving the trauma. This failure may be due to abnormally high levels of norepinephrine during REM sleep in PTSD patients. Their brains do not achieve the norepinephrine-free state necessary for emotional processing. The memory is replayed, but the emotion is not separated. The cycle repeats night after night, reinforcing the trauma rather than resolving it. Interestingly, a drug called prazosin, which blocks norepinephrine receptors, has shown promise in treating PTSD nightmares. By reducing norepinephrine activity during sleep, prazosin may allow the brain to finally process traumatic memories the way it should, separating the feeling from the fact. This pharmacological evidence supports the idea that norepinephrine suppression during REM is crucial for emotional processing. REM sleep may also play a role in fear extinction, the process by which a conditioned fear response gradually fades with repeated safe exposure. In animal studies, rats that are allowed to enter REM sleep after fear extinction training show better retention of the extinction memory, meaning they are less fearful when tested later. Disrupting REM sleep disrupts extinction, leaving the animals more fearful. This suggests that REM sleep helps consolidate the new, safe memory, overriding the old fearful association. The relationship between REM sleep and emotional health extends beyond trauma. Studies have found that people with depression often have abnormal REM sleep, including shortened REM latency, the time it takes to enter the first REM period after falling asleep. Depressed individuals enter REM faster than healthy people, and their REM sleep is often more intense. Some researchers have speculated that antidepressants, many of which suppress REM sleep, work partly by modulating this process, though the exact mechanisms remain unclear. What seems clear is that REM sleep is not just about dreaming for its own sake. Dreams are the surface manifestation of a deeper process, the brain's nightly effort to make sense of emotions, to integrate them, to reduce their intensity, to move on. In a very real sense, REM sleep is the brain's way of taking the edge off life, of ensuring that yesterday's emotions do not tyrannize today. Restoring the brain to criticality. There's a concept in physics and complex systems called criticality. It refers to a state of a system where it is balanced at a critical point between order and chaos, stability and flexibility. A system at criticality can respond to small inputs with a wide range of outcomes, from tiny local changes to large cascading effects. This makes the system maximally responsive, adaptable, and capable of rich behavior. Surprisingly, this concept appears to apply to the brain. Neuroscientists have observed that the waking brain operates at or near criticality, a state where neural activity is poised between too much order, where everything is rigidly locked together, and too much chaos, where everything is random noise. At criticality, the brain can process information efficiently, respond flexibly to new inputs, and support complex cognition. But maintaining criticality requires balance, and wakefulness disrupts that balance. Every sensory input, every thought, every decision, every movement alters the brain's state. Neural connections strengthen or weaken. Networks become more or less coordinated. Gradually, over the course of the day, the brain drifts away from criticality. It becomes less efficient, less flexible, harder to use. This is where sleep comes in. Recent research, published in early 2024, suggests that one of the primary functions of sleep is to restore the brain to a state of criticality. Researchers at Washington University in St. Louis tracked the neural activity of rats continuously for up to 2 weeks, monitoring how the brain's dynamics changed with sleep and wakefulness. They found that during wakefulness, especially during active behavior, the brain's deviation from criticality increased. The longer the rats stayed awake, the further they drifted. During sleep, the deviation decreased, bringing the brain back toward the optimal critical state. The effect was most pronounced during non-REM sleep, and it correlated with the amount of prior wakefulness. The more the rats had been awake, the more their brains had deviated from criticality, and the more restorative the sleep needed to be. The researchers measured this using a concept called neural avalanches, patterns of activity that spread through the brain. At criticality, avalanches come in all sizes, from tiny local events to large coordinated cascades. Away from criticality, the distribution changes. The brain becomes biased toward either small, insignificant avalanches that carry little information or large, unchecked avalanches that lack specificity. Neither is ideal for computation. Sleep, it seems, rebalances the system. It allows the brain to reset, to recalibrate, to return to the sweet spot where it functions best. This provides a computational explanation for why sleep is necessary. The brain is a machine that processes information, and like any machine, it requires maintenance. Wakefulness is productive, but costly. It uses the machine, and in doing so, it wears it down, knocking it out of its optimal operating state. Sleep is the maintenance window, the time when the machine is taken offline, tuned up, and returned to peak performance. This perspective integrates many of sleep's known functions. Memory consolidation, for instance, is part of the tuning process. Strengthening important connections and weakening irrelevant ones helps optimize the network. Glymphatic clearance removes the metabolic waste that would otherwise interfere with neural signaling. Emotional processing prevents the amygdala from becoming hyperactive and the prefrontal cortex from losing control. All these processes contribute to restoring criticality. The criticality framework also explains why sleep deprivation is so impairing. Without sleep, the brain does not just fail to consolidate memories or clear waste, though it does those things too. It also drifts further and further from its optimal computational state. Thinking becomes sluggish, attention wavers, creativity dries up. The brain still functions, but it functions poorly, like an engine running on bad fuel with clogged filters. Interestingly, the study found that not all waking is equal. Active, engaged waking, where the rats were exploring and interacting with their environment, caused a greater deviation from criticality than passive waking in the dark. This suggests that the intensity of waking experience matters. A day spent thinking hard, learning new things, and engaging with the world may require more restorative sleep than a day spent quietly resting. This may explain individual differences in sleep need. Some people function well on 6 or 7 hours of sleep, while others need 9 or 10. Part of this variation is genetic, but part may reflect differences in how intensely people use their brains during waking hours. Someone whose day is cognitively demanding may need more sleep to restore criticality than someone whose day is routine and undemanding. The criticality framework is still relatively new, and not all neuroscientists agree on its details, but it provides a compelling unifying hypothesis for sleep. Sleep is not about any one specific function. It is about restoring the brain to a state where all its functions can operate optimally. Criticality is the set point, the target state that the brain seeks to return to each night. When sleep eludes us. If sleep is so essential, what happens when it is denied? The answer is grim and multifaceted. Sleep deprivation is one of the most reliable ways to impair human performance across nearly every domain, from the physical to the cognitive to the emotional. Cognitive effects appear quickly and dramatically. After just one night of total sleep deprivation, attention and vigilance plummet. People become slower to react, more prone to errors, and less able to sustain focus on tasks. Lapses of attention become frequent, brief moments where the brain disengages from the task at hand, sometimes for just a second or two, but long enough to miss critical information or make dangerous mistakes. These lapses are unpredictable and can occur at any time, making sleep-deprived people dangerous when driving or operating machinery. The psychomotor vigilance task, a simple test where participants must press a button as quickly as possible when a stimulus appears, reveals these deficits clearly. After 24 hours without sleep, reaction times increase dramatically, and lapses, failures to respond within a reasonable time frame, multiply. After 48 hours, performance becomes catastrophically impaired. People sometimes fall into micro-sleeps, brief episodes where they lose consciousness for a few seconds while ostensibly remaining awake. Their eyes might remain open, but their brain has checked out. Memory suffers profoundly and in multiple ways. First, sleep-deprived people struggle to encode new information. Their hippocampus shows reduced activity in response to new stimuli, as if the structure is too exhausted to do its job. Brain imaging studies using functional MRI have shown that after a night without sleep, hippocampal activation during learning tasks drops by up to 40% compared to the well-rested state. Even if a sleep-deprived person manages to pay attention and take in information, that information often fails to stick. Second, even if some information is encoded, it remains fragile because it cannot be properly consolidated. Without sleep to replay and strengthen the memory, it fades quickly. Studies comparing people who learn material and then sleep versus people who learn and stay awake find massive differences in retention. After a week, the sleepers remember 2 to 3 times as much as the sleep-deprived group. Third, and perhaps most disturbing, sleep-deprived people are more likely to form false memories, confidently recalling events that never happened. The brain, struggling to make sense of incomplete or poorly encoded information, fills in gaps with plausible fabrications. These confabulations feel real. The person has no awareness that their memory is false. Studies have shown that after a night of total sleep deprivation, people are twice as likely to identify a face they have never seen as familiar, demonstrating how sleep loss degrades the fidelity of memory. The prefrontal cortex, which normally guides memory encoding and retrieval and provides executive control over cognition, also shows markedly reduced activity after sleep deprivation. This region is crucial for planning, decision-making, impulse control, and working memory. When it is impaired, people become more impulsive, less able to think through consequences, and more likely to make poor decisions. They lose the ability to hold multiple pieces of information in mind simultaneously, making complex reasoning nearly impossible. Working memory, the mental workspace where you temporarily hold and manipulate information, collapses under sleep deprivation. Try to do mental arithmetic, follow a complicated set of instructions, or keep track of a conversation with multiple threads when severely sleep deprived, and you will quickly find yourself lost. the information simply will not stay in mind long enough to work with. Emotional regulation collapses without sleep, often before cognitive deficits become obvious. The amygdala, the brain's alarm system and emotional processing center, becomes hyperactive. Studies have shown that after 35 hours of wakefulness, the amygdala's response to negative images increases by up to 60% compared to the well-rested state. It becomes hypersensitive, overreacting to stimuli that would normally provoke only mild responses. At the same time, the prefrontal cortex, which normally dampens amygdala activity and provides rational control over emotional responses, loses connectivity with the amygdala. The result is emotional volatility. Sleep-deprived people overreact to minor frustrations, feel more anxious and irritable, and have difficulty recovering from negative emotions. A small setback that would be brushed off when well-rested can feel catastrophic after a sleepless night. An ambiguous social interaction that might go unnoticed becomes a source of worry and rumination. This is not just subjective. Brain imaging shows the changes in real time. The amygdala lights up intensely on fMRI when a sleep-deprived person views mildly upsetting images. The prefrontal cortex, meanwhile, remains muted, failing to exert its regulatory influence. The brain, in effect, reverts to a more primitive mode of emotional processing, dominated by the limbic system without the moderating influence of higher cognitive centers. REM sleep deprivation specifically impairs emotional memory processing. When people are selectively deprived of REM sleep while being allowed to have NREM sleep, they show impaired ability to control intrusive memories. A study published in late 2024 found that after REM deprivation, people could not suppress unwanted memories as effectively. The right dorsolateral prefrontal cortex, a region critical for memory suppression, showed reduced activity. At the same time, the hippocampus showed increased activity during attempts to suppress memories, indicating that the suppression mechanism had failed. The memories intruded despite efforts to keep them at bay. This has obvious implications for mental health. Many psychiatric disorders involve intrusive thoughts and memories. Depression, anxiety, PTSD, obsessive-compulsive disorder, all feature unwanted mental content that the person cannot control. If sleep, particularly REM sleep, is essential for maintaining control over memory and emotion, then chronic sleep disruption may be both a symptom and a cause of these disorders. Chronic sleep deprivation, the kind experienced by shift workers, new parents, medical residents working extended shifts, or people with untreated sleep disorders like sleep apnea, has even more severe and insidious consequences. Studies tracking people who sleep less than 6 hours per night for extended periods show cumulative impairments in cognitive function that build over weeks and months, creating deficits that the person may not even recognize. After 2 weeks of sleeping 6 hours per night, people perform as poorly on attention tasks as people who have been totally sleep-deprived for 2 nights. Their reaction times slow, their error rates climb, and their ability to sustain attention collapses. Yet the chronically sleep-deprived people often do not feel as impaired as the acutely sleep-deprived. Their subjective sense of sleepiness plateaus after a few days, even as their performance continues to deteriorate. They are, in essence, too sleep-deprived to realize how sleep-deprived they are. This creates a dangerous situation where people believe they are functioning adequately when in fact their performance is severely compromised. This phenomenon helps explain why chronically sleep-deprived individuals, shift workers, truck drivers on long hauls, medical residents on call, may underestimate their impairment and engage in risky behaviors like driving while dangerously tired. They have adapted to feeling tired, but they have not adapted functionally. Their brains are still impaired. The risk of accidents increases dramatically. The physical health consequences of chronic sleep loss are profound and extend across nearly every organ system. Sleep deprivation is associated with increased risk of obesity, type 2 diabetes, cardiovascular disease, weakened immune function, certain cancers, and shortened lifespan. These are not small effects. Large epidemiological studies tracking hundreds of thousands of people over years have found that chronic short sleepers, those consistently getting less than 6 hours per night, die younger on average than people who sleep 7 to 8 hours. Some of these effects are mediated by hormonal changes that occur with sleep loss. Sleep deprivation increases levels of ghrelin, a hormone produced by the stomach that stimulates appetite and promotes food intake. At the same time, it decreases levels of leptin, a hormone produced by fat cells that signals satiety and reduces hunger. The result is a perfect storm for weight gain, increased hunger, particularly for high-calorie, high-carbohydrate foods, combined with reduced feelings of fullness. Sleep-deprived people tend to eat more overall, snack more frequently, and gravitate toward junk food. Over time, this leads to weight gain and obesity. Sleep loss also impairs glucose metabolism and insulin sensitivity. Even a few nights of restricted sleep, down to 4 or 5 hours per night, can reduce the body's ability to respond to insulin, the hormone that regulates blood sugar. In studies where healthy young people are sleep restricted for less than a week, their glucose tolerance drops to levels typically seen in prediabetes. If this becomes chronic, it sets the stage for type 2 diabetes. The cardiovascular system suffers as well. Chronic sleep deprivation is associated with hypertension, irregular heart rhythms, increased inflammation, and higher risk of heart attack and stroke. The mechanisms are multiple. Increased sympathetic nervous system activity, keeping the body in a state of stress, elevated cortisol levels, inflammatory markers like C-reactive protein rising, and endothelial dysfunction where the lining of blood vessels becomes damaged. Sleep, particularly deep slow wave sleep, normally allows the cardiovascular system to rest and recover, with heart rate and blood pressure dropping to their lowest levels. Without adequate sleep, the system never fully relaxes, remaining in a state of chronic activation that damages it over time. Perhaps most alarmingly for our long-term future, chronic sleep deprivation is linked to accelerated brain aging and increased risk of neurodegenerative disease. As discussed earlier, insufficient sleep impairs glymphatic clearance, the brain's waste removal system, allowing beta amyloid and other toxic proteins to accumulate. Over years and decades, this accumulation contributes to the development of Alzheimer's disease and other forms of dementia. Studies have found that people who chronically sleep less than 7 hours per night have higher rates of Alzheimer's disease later in life, even after controlling for other risk factors like genetics, education, and lifestyle. The relationship appears to be dose-dependent, meaning the less you sleep, the greater your risk. Those who sleep less than 5 hours per night face particularly elevated risk, with some studies suggesting their risk may be double that of people who sleep 7 to 8 hours. Brain imaging studies provide visual evidence of this damage. Even in middle age, decades before dementia symptoms would appear, poor sleepers show accelerated brain aging. Their brains show more atrophy, meaning the brain tissue is shrinking faster than it should. The cortex becomes thinner, losing gray matter. The ventricles, fluid-filled spaces inside the brain, enlarge as surrounding tissue recedes. The hippocampus, so critical for memory, shows particularly pronounced shrinkage in people with chronic sleep problems. A study published in 2024 followed individuals with mild cognitive impairment, a condition that often precedes Alzheimer's disease, and found that those with greater sleep deprivation showed faster progression of hippocampal atrophy and reductions in total brain volume over just 6 months. The connection was clear. Poor sleep was not just correlated with brain aging, it was predicting the rate at which the brain deteriorated. White matter, the bundles of nerve fibers that connect different brain regions, also suffers. Studies using diffusion tensor imaging, a technique that maps the integrity of white matter tracts, have found that chronic short sleepers show more white matter hyperintensities, lesions that appear as bright spots on brain scans and indicate damage to small blood vessels and the surrounding tissue. They also show reduced fractional anisotropy, a measure of white matter structural integrity. Lower fractional anisotropy means the white matter is less organized, less efficient at transmitting signals between brain regions. These changes are not benign. White matter hyperintensities and reduced fractional anisotropy are strong predictors of future cognitive decline, stroke, and dementia. They represent silent brain injuries, damage accumulating beneath the surface that will eventually manifest as noticeable impairment. The fact that they appear more frequently in poor sleepers suggests that sleep deprivation is actively damaging the brain's infrastructure. The mechanisms are multiple and interconnected. Without adequate glymphatic clearance, toxic proteins accumulate. Without proper memory consolidation, synaptic connections fail to strengthen or are inappropriately pruned. Without the restorative processes of deep sleep, neurons cannot repair damage from oxidative stress. Without REM sleep, emotional regulation fails, and chronic stress hormones like cortisol remain elevated, which itself is toxic to the hippocampus over time. The brain ages faster when sleep is inadequate, like a machine running without maintenance, gradually breaking down. Recovery from sleep deprivation is possible but not immediate or complete. After a night or two of poor sleep, people can bounce back relatively quickly with 1 or 2 nights of good rest. Those subtle deficits may persist for several days. After chronic sleep restriction, recovery takes longer. Some studies suggest that 2 recovery nights are insufficient to fully restore hippocampal connectivity and memory performance after just 1 night of total sleep loss. The brain seems to need time to repair, to clear the backlog of metabolic waste, to re-optimize its networks, to rebuild what was damaged. More concerning, some damage may be irreversible. While acute sleep deprivation is mostly recoverable, chronic sleep deprivation, particularly if it lasts for years, may cause permanent changes. The accelerated brain aging seen in chronic short sleepers may not be fully reversible, even if sleep improves later in life. This means that sleep habits in middle age may determine brain health in old age, with consequences that only become apparent decades later. This means that sleep debt is real, and it accumulates in ways we are only beginning to understand. You cannot simply make up for lost sleep on the weekends after sleeping poorly all week. While weekend catch-up sleep is better than nothing, it does not fully compensate for the cumulative damage of chronic restriction during the work week. The brain needs consistent, adequate sleep every night, not periodic binges of recovery sleep interspersed with prolonged deprivation. The lesson is clear. Sleep is not optional. It is not a luxury. It is a biological necessity, as fundamental as food or water. Deprivation brings impairment, and that impairment extends far beyond simple tiredness. The brain and body cannot function properly without adequate sleep, and the deficits that accumulate can have lasting consequences. A polyfunctional mystery unfolding. For decades, researchers sought a single answer to the question of why we sleep. Is it to conserve energy? To consolidate memories? To clear metabolic waste? To process emotions? To restore synaptic balance? To optimize brain dynamics? The answer, we now understand, is yes, all of the above. Sleep is polyfunctional. It serves many purposes simultaneously, and these purposes are deeply interconnected. You cannot tease them apart into independent functions because they support and reinforce each other. Memory consolidation benefits from glymphatic clearance because removing waste products improves synaptic function. Emotional processing depends on the unique neurochemistry of REM sleep, which in turn supports memory consolidation by allowing synaptic changes that cannot occur in the presence of norepinephrine. Restoring criticality involves optimizing synaptic weights, which overlaps with memory consolidation. The brain cannot clear waste efficiently without the synchronized oscillations of slow-wave sleep, which are also critical for memory transfer from hippocampus to cortex. Sleep is not a collection of separate functions happening in parallel. It is an integrated process, a state that the brain enters because it allows a suite of essential activities that cannot happen or happen poorly during wakefulness. This realization represents a major shift in how sleep science thinks about its subject. The polyfunctional view also helps explain why sleep is so conserved across species. Different animals may emphasize different aspects of sleep's functions depending on their ecology and life history, but the core necessity remains. A species with a simple brain may benefit primarily from the energy conservation and niche separation aspects of sleep, while a species with a large, complex brain benefits additionally from memory consolidation, emotional processing, and computational optimization. Sleep evolves and adapts, but it persists because it solves multiple problems at once. This also explains the diversity of sleep patterns in nature. Dolphins sleep with one hemisphere at a time because they cannot afford to stop breathing, but they still need the restorative functions of sleep. Migratory birds reduce their sleep during migration, but compensate with more sleep before and after. Some semilparous marsupials, animals that reproduce once and then die, reduce sleep during their mating season, apparently prioritizing reproduction over long-term brain health because they will not survive long enough to pay the long-term costs. These variations show that sleep is flexible, but they also show that its functions are real. Animals sacrifice sleep only when immediate survival demands it, and they pay a cost for doing so. Recent advances in sleep science have been nothing short of revolutionary. The discovery of the glymphatic system reframed sleep as a time of active brain maintenance. The identification of criticality as a target state provided a unifying computational framework. The elucidation of memory replay mechanisms and the role of specific oscillations in coordinating consolidation revealed the intricate choreography underlying learning. The understanding of REM sleep's emotional functions offered insight into mental health and trauma. These discoveries have been enabled by new technologies. High-density EEG and intracranial recordings allow researchers to track neural activity with unprecedented precision. Optogenetics, a technique that uses light to control genetically modified neurons, allows specific sleep stages or neural populations to be manipulated, revealing causal relationships rather than mere correlations. Advanced imaging techniques like two-photon microscopy allow real-time visualization of synapses and glial cells in living animals during sleep. The pace of discovery is accelerating. Just in the past few years, studies have revealed how sleep affects the gut microbiome, how the microbiome in turn affects sleep, how sleep modulates immune function at the molecular level, how chronic sleep disruption accelerates aging in cells, and how sleep interventions might be used therapeutically to treat everything from PTSD to Alzheimer's disease. Yet for all we have learned, mysteries remain. We still do not fully understand the molecular mechanisms that generate the need for sleep, the homeostatic pressure that builds during waking and dissipates during sleep. We do not know exactly how the brain decides which memories to consolidate and which to forget. We do not know why humans, alone among primates, have such long and consolidated sleep periods, or why we dream more vividly than most other animals. We do not know if artificial intelligence systems, which also process information, would benefit from something analogous to sleep, or if the need for sleep is unique to biological brains with their specific biochemical and metabolic constraints. What we do know is that sleep is not a simple state of inactivity. It is a complex, dynamic, essential process that has been shaped by hundreds of millions of years of evolution. It is as fundamental to life as breathing or eating, and it touches every aspect of health, cognition, and well-being. Closing reflection. We began tonight with a question. Why do we sleep? Why do we spend a third of our lives unconscious, vulnerable, disconnected from the world? Now, as we near the end of this exploration, perhaps you see that the question was never going to have a single, simple answer. Sleep is not one thing. It is everything the brain and body need to do that cannot be done while awake. It is a state of such fundamental importance that evolution has preserved it across hundreds of millions of years, despite the obvious costs and dangers it imposes. Sleep is when your brain replays the day, moving memories from temporary storage to permanent archives, integrating new knowledge with old, extracting patterns and meaning from experience. The hippocampus, that seahorse-shaped structure deep in your temporal lobe, fires in precise sequences, teaching the cortex what you learned, strengthening the connections that matter, letting the irrelevant fade. Night after night, your brain reviews and refines what it knows, building the continuity of your life, the sense that you are a person with a history, an identity, a collection of knowledge and skills that define who you are. Sleep is when your brain washes itself, when cerebrospinal fluid surges through the spaces between cells, flushing out the metabolic waste that accumulated during waking hours. Proteins like beta amyloid, which would otherwise clump into the toxic plaques of Alzheimer's disease, are swept away in this nightly cleansing. The glymphatic system, discovered only recently, works tirelessly through the night, ensuring that the brain does not drown in its own metabolic byproducts. Without this process, without this regular maintenance, the machinery of thought would gradually clog and fail. Sleep is when your neurons recalibrate themselves, restoring the delicate balance of excitation and inhibition, the criticality that allows for flexible, efficient computation. Waking experience pushes the brain away from this optimal state, biasing it toward patterns that are too rigid or too chaotic. Sleep brings it back to center, to that sweet spot where the brain can respond adaptively to whatever comes next. It is a reset, a return to baseline, a restoration of the brain's ability to think clearly and respond appropriately. Sleep is when your emotions are processed, their intensity softened, their lessons extracted and integrated without the overwhelming force of the original feeling. REM sleep, that strange state where your brain is active, but your body is paralyzed, where dreams unfold in surreal narratives, provides a neurochemical environment where emotional memories can be reactivated without the stress response that normally accompanies them. The norepinephrine falls silent. The amygdala and hippocampus converse. Slowly, night by night, the sting of difficult experiences fades, leaving the memory intact but the emotion manageable. This is overnight therapy, the brain's way of helping you move on, of preventing the past from tyrannizing the present. Sleep is when your body repairs tissue, balances hormones, strengthens immune defenses, and resets the myriad physiological systems that keep you alive and healthy. Growth hormone pulses through your bloodstream during deep sleep, promoting tissue repair and maintenance. Your immune system ramps up production of infection-fighting cells. Your cardiovascular system gets a break, with heart rate and blood pressure dropping to their lowest levels. Inflammation subsides. The body uses this time away from waking demands to heal, to prepare, to restore what was depleted. In a very real sense, sleep is what allows you to be you. It maintains the continuity of your identity, the integrity of your memories, the stability of your emotions, the sharpness of your mind, the health of your body. Without it, you fragment. Memories become hazy and unreliable. Thinking becomes sluggish and error-prone. Emotions become volatile and overwhelming. Physical health deteriorates. The machinery of self begins to break down piece by piece, system by system. And yet, despite its absolute necessity, despite the fact that we spend a third of our lives doing it, sleep remains mysterious in many of its details. We do not yet have a complete understanding of all its mechanisms, all its functions, all the ways it varies across species and individuals. Science is still uncovering how deeply sleep is woven into every aspect of biology, from the molecular machinery inside individual cells to the social dynamics of entire societies. New discoveries are made each year, each one revealing another layer of complexity, another reason why this nightly vanishing is not a waste of time, but an essential investment in tomorrow. What we have learned, though, is enough to know that sleep is not negotiable. It is not a luxury to be sacrificed when life gets busy. It is not a sign of weakness or laziness. It is a biological necessity, as fundamental as food or water or air. Deprivation brings impairment, and that impairment extends far beyond simple tiredness. The brain and body cannot function properly without adequate sleep, and the deficits that accumulate can have lasting, sometimes permanent consequences. So tonight, as you prepare to sleep, perhaps you can do so with a new appreciation. You are not simply shutting down for a few hours of blank nothingness. You are allowing your brain to enter a state that has been perfected over hundreds of millions of years of evolution, a state where countless processes unfold in precise coordination, each one essential to your health and your humanity. You are giving your hippocampus time to replay the day and teach the cortex. You are opening the glymphatic floodgates to wash away toxic waste. You are allowing your neurons to reset to criticality. You are processing emotions in the safety of REM sleep. You are repairing your body and rebalancing your hormones. You are doing what every animal with a brain must do. You are sleeping. Sleep is not time lost. It is time invested in tomorrow. If you found tonight's exploration helpful or interesting, if it gave you a moment of calm or a spark of curiosity, please consider liking this video and subscribing to Quiet Science channel. It helps others discover these gentle conversations about the mysteries of science, one peaceful night at a time. Thank you for joining me tonight. Rest well, and let sleep do its quiet, essential work. Until next time, good night.